All right. I forgot the sheets in my office, so I'll make sure to scan those later, the ones that you guys worked on in groups on Wednesday. But who had the question referring to figure 2? Otherwise, I start to pick on people at random. Okay, Abby, why don't you give it a stab? Give it a stab, anyway.
Figure 1. Figure 1 part.
It wasn't me.
Okay, well.
Give it a stab anyway. Oh, sorry, it's supposed to be up here. My apologies. Go ahead.
Figure 2 is them all at 40 degrees Celsius. 20 degrees Celsius, right?
Right, that's the major difference between the two of them. So what are the major, if you're looking at this, just describe the results as is right now. Don't even compare back to figure 1 yet.
Oh, okay, well, for figure A, which is the gram-positive bacteria, it looks like that works better, right? Like at killing or like removing the bacteria.
What works better?
Both the plain soap and antibacterial soap.
Okay, so washing hands is better than not washing hands,
Yeah.
right? And Mom was right. Correct. Okay. Difference between those two conditions, though?
It's not really significant.
Yeah, I think that's the most important thing to say. There may be minor differences, but it obviously isn't significant, right? If you take into account those error bars, they overlap, which to me would say there's no difference between these two groups. Those differences could be explained simply by like experimental technique as much as they could be explained by real results or real experimental differences in the results here. Take a look at this in comparison to figure 1 real quick. So note that log decrease is about 1 to 2 logs in each of these conditions here. Compare that to this. Is there a difference with temperature? Does one temperature work better than the other in terms of removing bacteria, or at least leading to a decrease in bacterial numbers? Nicole, you're shaking your head no.
I guess I don't really see a big difference.
I don't either. It's not there, right? So does that mean that washing with 40 degrees Celsius water on your hands, is that going to make a difference in 22? And I think that maybe some people would say no. I would probably say no, but there's a caveat to this. Remember how this experiment was performed. So if you remember when we talked about this, these experiments here in 1 and 2 were done in a test tube. Incubation with soap in a test tube for 20 seconds at either 22 or 40 degrees Celsius. So the bacteria were in contact with that temperature for a certain amount of time. What that's saying is that at least the temperature doesn't have an effect on the death of the microorganism. What it doesn't tell us is in the context of hand washing, does that temperature have an effect on removing the bacteria off of hands at one of those two temperatures? So I just wanted to point out that caveat. That's one thing that they actually don't cover in this study is whether temperature actually makes a difference. And for those of you, I don't know if we could do this or not, but we're getting to independent projects in the lab in the next couple of weeks. This could be a potential question to ask, whether temperature actually does make a difference. Okay? Something to think about. All right, I want to move on to figure 3, and again, I apologize for not bringing these sheets, but if somebody remembers, what I want to do is somebody to explain for us what figure 3 is. Can somebody at least start with what are the x and y axes showing us? So that we can at least set up this particular experiment again. Go ahead, Paige.
Isn't it like how long it takes for the reaction of the the bacteria to the soap?
Yeah, more generally, just how long does it take the bacteria to die in the presence of the soap or the soap with triclosan? So here's time on the x-axis, y-axis again is a measurement of population. Again, this is just like what's being done in 1 and 2, nothing else added to it. So we've seen 4 figures now. This one is probably the most representative of how soap would be used in everyday life. or at least antibacterial soap would be used in everyday life. What I want you to do is take a minute, and just, you don't have to write this one down, but just think about what are the major take-home points. If you were to summarize the results of this study, what are the major take-home points? And put this in the context of the individual or the original question. The original question was, does washing hands with soap with triclosan, does that have an additional effect? Okay, I'll give you a minute to think about it. Friday. Friday. Well, it's due the time that you enter lab on Friday. For the morning class, it's due Thursday because that's when their lab time is. So I've given you guys the same number of days after you completed those experiments to do it. All right. General theme, summarize the results. What's the takeaway point here? Jaden?
It really doesn't matter.
It doesn't, does it? So, was the a follow-up question then? Was the FDA right to have banned triclosan from hand soaps? And I mean, a mixed answer, right? Because why do you say yes, Paige or Cassidy?
No, I said it doesn't really matter.
Oh, it doesn't really matter. Okay, Paige, go ahead, Cassidy.
I just think like if it doesn't make a difference, then why do you need a banner?
Sure, sure. Paige?
Like to me, I feel like because it was causing antibac resistance, and it's not actually helping to make your hands any cleaner.
Okay.
What's really the point in exposing yourself to like more resistant bacteria when your hands are going to be just as clean either way?
Okay, good. Zach?
Doesn't it cost like $1.3 billion a year to fund or something like that?
Yeah, that's a really good point too. So when you put into context the cost, like the amount of money that we pay extra for antibacterial soap, when there's no proof or no support that it actually has an additional benefit, I mean that's the kind of things that the FDA is supposed to protect us against, are those false claims. But when you consider the potential drawbacks, I think really the FDA's decision came down to the potential drawbacks from this in terms of potential antibiotic resistance, in terms of the allergies that have been associated with triclosan or some of these other products. Those outweigh the potential benefits. There are no benefits, at least from the perspective of hand washing. Okay? So the FDA, the end result of this, and this is just kind of one of many papers that led to this decision, 19 out of 20 different antibacterial products were outlawed as of last summer, 2016. The one that they still hadn't decided on were the quaternary ammonium compounds. And they were, companies that were using that as an antibacterial were allowed, they were being given another year actually to test and determine is there an additional effect. Another study question, if anybody wants to, because guess what? We've got antibacterial soap with benzalkonium chloride, which is a quaternary ammonium compound. We've also got soap in the lab that doesn't contain it as well, that you can use as a comparison. So, just getting you to think already about what's coming up down the road. Yay! I'm super excited. I truly am. I know. It's kind of like home. Only one of us is excited. Me. All right. Before we go to this, I wanted to pull up something on Blackboard. So, takeaway point from this. Will I ask you about this particular paper on the exam? Is it worthwhile going back and reviewing every single thing we've talked about with it? No. No, that's not the goal here. The goal is to think about how does the data inform our decisions. And to kind of keep in mind, I mean, we reinforce concepts here of counting bacteria, so ways to measure growth, and then looking at ways to control growth. Okay, so those are kind of the themes that I really want you to focus on. With that in mind, what we're going to do is a quick activity. And sorry for all the polls and stuff lately. I really apologize. I haven't been on top of my game. All right. So what you guys are going to see, if you go under today's lecture, actually, so we're on antibiotics number 1. What I'm going to start doing, I mentioned that I would do these good, better, or best questions, where I would pose a question and then I'll give 3 responses afterwards. One that I would consider to be like a 1 out of 3, a 2 out of 3, or a 3 out of 3 on a 3-point scale. Here's the first of those. We're actually going to do this one in class today. Not from the paper, but actually quite similar in concept. So here is the question. It says, analyze the data in the table, shown here. In the experiment, instead of using Serratia, they had individuals who coated their hands with spores of C. diff. Okay, right on the palm. And what they ended up doing then was testing how did a detergent-based product, which is the chlorhexidine washing, that's the brand name is Hibiclens, and then how did 3 different ethanol-based hand rubs, Purell, Endure, and Isogel, how did those perform in reducing the amount of spores on the hands of individual or these test subjects? Okay, so there are 3 things that I want you to do. This is really a 3-part question. 1, describe the general trend of the data, but then you also need to use some specific examples to support that trend that you've identified. Next, interpret the results. And then the last is identify any likely explanations for the findings. Okay? I'm going to give you 5 minutes. Write down your answers for this one, and then we'll walk through it afterwards. So I want this to be individual because this is a good test to see where are you at with this type of information right now. Okay? And this is up on Blackboard if you'd like to see it on your computer. Don't look at the answers just yet, though. It should be under Lecture 1, and it's towards the bottom, or Antibiotics Lecture 1. So keep scrolling. Oh, and while we're doing this, I'm going to come around real quick because I forgot to do that. If you have that sheet of antibiotics that I asked you to have with, just hold it up or show it to me on your computer. I'm just going to walk by real quick. All right, let's go over this. So I'm not going to ask for any complete answers right now. What I'm just going to ask for is individual pieces of answers, and anybody can feel free to contribute. Okay? And then the goal is to build this into a really good response. I'm not going to go over the ones that I've posted, but I will say what you'll find in there is that I've got the 3 different types of responses: a good, a better, a best. And in each one, I've bolded some phrases or terms that make, for example, a better different from a good, or that make a best different from a better response. Okay? And you can use that then to kind of self-assess where are you at as of right now. So let's just start with the basics. Describe the general trend. That's not a good start. Go ahead, Danica.
Is it that the alcohol-based hand rubs have a lower mean reduction than the normal hand rubs?
Okay. So alcohol-based hand rubs, we're just going to call the AHR. Why not? Have, what was the term you used? Lower?
Lower mean reduction.
Okay. So a couple of things that I like out of this answer right away. The comparison. That's really important to have in there. The one term I might pick on here would be this one, only because we're looking at reductions, so a lower reduction can sometimes be confusing. And I would understand what you mean, but I'm just trying to get us to be specific. Is there another way we could say that instead of lower? Smaller would probably be a better word here. I think that would be a more fitting word for this one. Anybody have anything different than this? Okay. Examples. What are examples of that? So somebody pick out for me one example that fits this exact trend. Go ahead, Paige.
The way I have written it is that the chlorhexidine washing was superior in C. diff score reduction. So if you compare like the chlorhexidine washing to the isogel, you can see the 0.9 to the 0.1.
Good. And that's what I want. That's really, when I'm saying pick out a specific example, that's saying like report the results, report the data. So Paige has picked out here, chlorhexidine, the mean reduction was 0.9 logs. And again, this is on a log-based 10 scale. This is equivalent, though I wouldn't expect you to say this, it's equivalent to about a 90% reduction, 80 to 90%, whereas the isogel, only a reduction of 0.11. Okay, so that's actually like 10% or less than 10%, pretty small difference. Another, and you could pick out any of these comparisons, the chlorhexidine to Endure, the chlorhexidine to Purell. Maybe as it's showing at least the minimal difference, I might say chlorhexidine, the mean reduction was 0.9, and the best performing of all of the hand sanitizers, the Endure, only had a log decrease of 0.37. To put that into percentages real quick for you, that's only like 50%. And we talked about last week on Wednesday how 50% is not a difference really when it comes to the microbial world. Did anybody focus on p-values for this to support their evidence? You can. You don't have to, but that sometimes can add some sometimes can add some strength to the argument. Sarah?
What is the p-value?
Yeah, so the p-value is a measurement of statistical significance. In this one, the test, I don't know if anybody is familiar with Wilcoxon rank sum. Is that something that gets talked about in statistics here? I didn't think so. Basically, the test is like you would take what's the reduction for all the people that were treated with chlorhexidine versus all the people that were treated with one of those other, or versus unwashed hands, sorry. You then rank the log reduction of both of those compared to alcohol. 70% alcohol does nothing. Nothing at all. And guess what? 70% is about the concentration in most of these hand sanitizers. So maybe work really well to reduce vegetative or living or bacteria that are actively growing, but those that are dormant like a spore doesn't do a thing to them. What's another explanation? I mean, it's kind of something that's coupled with that a little bit. Think of the process. What, go ahead, Sid.
Did you say that like washing your hands is like a longer process to like fulfill the action than it is to just put hand sanitizer on your hands?
I wouldn't disagree with that. And actually, there's a longer period of contact time during the hand washing than like those hand sanitizers usually dry out so quickly that the amount of ethanol or the time of exposure is too low to actually have an effect. I would not disagree with that argument. One other one. What's different in using a hand Cassidy?
I was just wondering, would it make a difference because of like the amount of time, because the spores can like just like sit in the alcohol for so long?
Yeah, and I think that's kind of what Sid is hitting at too. I wouldn't disagree with that argument at all.
But like if, not that it's like likely, but if you put your hands in like hand sanitizer for like 2 hours, would it kill that endospores?
I don't know. My guess is no. It'd probably kill a lot else. It'd probably cause your hands to crack a lot.
Yeah.
But
I should know.
when we wash hands, what are the last two things we do after soaping up our hands? Rinse and dry. When we put on hand sanitizer, do we rinse or dry? No. I think that might be the other part of the explanation. There's actually a mechanical removal in the hand washing experience. That's a degerming event. And that could actually remove the spores and wash them down the drain where they can then be treated by a wastewater facility. Whereas those spores, putting on hand sanitizer, they stay there. Not enough alcohol to kill them, so they stay viable. Yeah.
Like hand dryers, though. Or is it just the rinsing?
I don't know. That's a great question. So, I mean, that's kind of a different question, right? That one would almost be like, does washing with hand drying work better than washing with and then drying with a towel afterwards? I don't know. That's a really good question. Would spores be able to survive the heat of a hand dryer? For the amount of time that you're sitting there? Probably. Spores are pretty heat resistant. I don't know. I have no clue. That's a great question. All right. Does this help a little bit to kind of demystify what kind of responses we're looking for? Right? At least is this a good start?
Yeah.
So we'll continue to do this. We'll do one on Wednesday. We'll do one on Friday. And then after the exam, we'll continue to do this with other ones. We've only got about 10 minutes left, but what I wanted to do was at least to get started talking about antibiotics. Now, I've put up a key of what I think the responses should be for the different antibiotics that you guys put your list together for, so feel free to compare it. It's in the same exact location on Blackboard. It's just I've included a second file below the actual table that you filled out. But rather than us taking the time to log on to Socrative, what I'm going to do is I'm just going to pull up this quiz. I'm going to give you a few seconds to think about your answer, and then we're going to go ahead and answer these, because I want to use this as a way for us to consider what are the roles of these antibiotics. Probably the simplest thing to say, antibiotics target proteins. There are a couple of exceptions, but most target proteins. And they inhibit some process within the cell, some metabolic process. Okay. So here's the first one. Which of these antibiotics directly inhibits translation? Is it the beta-lactams, the quinolones, the acetotracin, or the aminoglycosides? And feel free to use the sheets that you've got with you. This is kind of a way to check, if you have those answers correct or not. So I'll give you a second to think about it. All right. So which one targets translation directly? B, the quinolones? D, oh, sorry. Aminoglycosides. Who agrees with that one? Okay. Aminoglycosides. What's the example of the aminoglycoside that's on the worksheet that I gave you? Neomycin is the main one. And I will point out on Wednesday for you guys, what of the information on there do I really want you to know? Largely, to summarize, it is the class, the target, the protein target, and the process that's inhibited. There are a couple of exceptions to this, and I will make sure to note those for you. I'll probably just put together a little video at the end of today to let you know what not to worry about. Okay. How about these other ones? What do beta-lactams target? Or what is the process that they target? Cell wall synthesis. We'll cover these in greater detail. But there were two examples on that sheet. The one that I gave you was penicillin. What is another example of a beta-lactam? Sarah? Yeah, ampicillin. So any of those that end in like a cillin are primarily beta-lactams. Cephalosporins are other examples of this family, and then carbapenem, which is kind of like a last resort drug right now, is also a member. What about the quinolones? What do they target? Specifically DNA replication? Yeah, so they target one of the terms that I asked you guys about on that concept map was gyrase. And gyrase is the enzyme that's involved in actually releasing supercoiling, or sometimes you get replication where the two strands of DNA are linked at the end. Gyrase resolves those links. And so specifically the quinolones inhibit that. Paige?
Another term for like the low laxicin or whatever, the one that you had on
Yeah.
was fluoroquinolones.
Fluoroquinolones, yes. There's really not much of a difference. It's the presence of fluorine. So quinolone and fluoroquinolone, they function very much the same exact way, and actually we will consider them as a single class of antibiotic. Yeah. How about bacitracin? What's that? No, I didn't hear what you said. Did you say what about it? Everything, sorry. What does it target? What was that, Cassidy? Non-ribosomal proteins? That's true to an extent, yeah. I mean, that's kind of a broad definition. Cell wall synthesis, particularly, this one is what we would consider to be a narrow spectrum drug. We'll come back to that a little bit later. Okay. Now, what I'm saying might be a little bit different than what the book says. We're going to go off of what I say, not because I'm necessarily always right, but I want to go off of what I'm saying because I want us all to have a common language on this one. Okay. Some people would consider bacitracin, for example, to maybe be a broad spectrum. It's a qualitative measurement rather than a quantitative one. All right. Let's go to the next one. Discard. Which of the following is not part of triple antibiotic ointment? Neomycin, tetracycline, bacitracin, polymyxin B. Yeah. What's another name for a triple antibiotic ointment? A brand name. Neosporin. So neomycin is definitely in there. Neomycin is definitely in there. That one is a broad spectrum drug. What?
How do you know that?
I think it was on the mini lecture. Did I mention it on there?
Yeah.
Did I mention it on the mini lecture that Neosporin, bacitracin, and polymyxin are? If not, I meant to bring it up. I don't know. Because so neomycin definitely is, bacitracin definitely is, and then polymyxin is as well. Bacitracin and polymyxin are what we would classify as being narrow spectrum antibiotics. What do we mean by that term? So if we were to define a narrow spectrum antibiotic, what makes it different from a broad one? Jaden? Yeah, and typically I would define this as like if they target only the gram positives or only the gram negatives, I would consider it to be a narrow spectrum drug. Okay, Sydney.
On the table,
Yes, this is one.
like if it's a little bit in one of them.
I think bacitracin is inhibiting largely gram positives, and it describes polymyxin as inhibiting gram negatives. I go based off of that definition, and in part it's because, think of it this way, bacitracin can inhibit some gram negatives. I don't disagree with that. But it targets the cell wall. Most gram negatives have what structure to inhibit their cell wall from being targeted by bacitracin? They've got an outer membrane, which is going to protect. So, Cindy, that's one of the examples where I want to say, let's talk a common language on this one. Okay, I would largely consider it narrow spectrum. Jaden. Already posted. I did it this morning. So where you guys picked up this particular table, there's another file right under it labeled key. Yep. Yes. Now, do I want you to know the spectrum of these drugs? No. I don't, because what I say may be different than others. I may ask you a question on the exam, something where I define one as being narrow spectrum, and then you tell me what that means or what are the implications of that in terms of its use. But I am not going to ask you about the spectrum based on the name of the drug. Okay? Polymyxin B, another narrow spectrum, specifically targets outer membrane. It actually leads to the destabilization of the outer membrane on those gram-negative bacteria. So this is one that doesn't have a protein target. It actually is just going to lead to leakiness of that outer membrane on those gram-negative cells. May I just finish with one more? Sure, we'd love to. And this is where we're going to pick up on Wednesday. Polymyxin B is only used topically. Why? So, you don't have to answer this one right now. What I want you to think about is the term D. I'm not saying is or is not the right answer, but think of therapeutic index and what does that actually mean? Okay, and what goes into determining the therapeutic index? That's where we will pick up on Wednesday. Because I've run out of time, I'll put together just a short video to recap the major points from today, let you know of a couple of other things that you need to think of coming up into this week. Other than that, I'll see you guys later. Have a great couple of days. Jasmine. Test is next week on Monday. So we're going to do, so I'm going to hold a review session on Thursday. Haven't decided yet. Fill out the doodle poll that I sent you guys, or that's up on, there's a link for it on Blackboard.